Okay, so we're talking about adaptations to resistance training, and we're talking about the signals, the intracellular signals that are activated in response to in response to resistance training, and we want to talk about the variety of ways that we can get adaptations to those. Now, different stimulus activate different signaling molecules, but some of the same signaling molecules are affected in different ways. And this is, from a big picture standpoint, this is an important thing to understand from a molecular biology standpoint. So that when you're reading some sort of article that's talking about mTOR activation, and you're saying, no, I learned that mTOR activation is activated by insulin signaling through AKT. You need to realize that there's multiple ways that you can affect the same protein within a cell, and that there's like redundancies and there's varying levels of control within a cellular system within the response to a particular stimulus. So when you see papers that are discussing new mechanisms or new increased understanding of the way certain things are regulated within the cell, this is part of it, but it by no means is comprehensive in terms of everything that goes into regulating mTOR. Does that make sense? So it's really important to understand that, and clearly this discussion and this summary figure here demonstrates very clearly that there are multiple ways to regulate one important intracellular signal, and that depending on how you stimulate or the signals that you give a system, you can try and optimize those, the activation of those types of things. So the central protein that we are discussing currently is mTOR, or in its active form, it has those other proteins combined with it, and it makes what's called the mTOR complex 1, or mTOR1. And we said this is central to a lot of different downstream anabolic signals. And that's pretty much the thing that you need to, it's not the only thing you need to understand about mTOR, but it's in that anabolic category. So we don't expect mTOR to be activated during exercise. We expect mTOR to be activated by anabolic signals. mTOR may be related to adaptations that allow us to use energy more efficiently, like turning on PGC1 alpha, which turns on mitochondrial biogenesis. But it doesn't necessarily, mTOR isn't involved in the actual energy production when we're engaged in exercise. All right? So we need to think of it as this really important anabolic signal. Okay? And it can be activated in a bunch of different ways. Now, the other concept that's a basic molecular biology concept that can be a little bit tricky to wrap our heads around is this idea that if we inhibit an inhibitor, then you activate the protein that's downstream of that inhibitor. So what we see here is a lot of these signals acting on this TSC2 Reb complex. Right? And what's happening there is that TSC2 kind of binds Reb and prevents Reb from activating mTOR. So Reb is an inhibitor of mTOR, or it suppresses the activity of mTOR. Inhibitor is kind of a strong word. You have to be careful with the way you use that, because it's as if you can't, there's no other thing that can stop mTOR, and if Reb is present, then there's absolutely no mTOR. You want to be careful using that type of language. So we'll just say that Reb suppresses the activity of mTOR, and when we suppress the activity of TSC2 or alter the relationship between TSC2 and Reb, then we can prevent Reb from inhibiting mTOR, which allows mTOR to be activated. Now, the downstream components of mTOR are many, but you want to think of them, really one of the more important ones that we want to think about in the context of this class is that it's a stimulator of protein synthesis. So it turns on protein synthesis. And when I say protein synthesis, I mean protein translation. So this, where the mRNA has been, the introns have been spliced out, the exons have been spliced together, the mRNA that has the code for the polypeptide to make the protein is ready to go. It just needs to get to the ribosome, go into the ribosome, and then have the ribosome do its thing, where the ribosome reads the mRNA chain 3 nucleic acids at a time, right? The tRNA comes in and adds the peptides 1 at a time, and then you have, that's the basic step in protein synthesis or protein translation, right? And in order for that to happen, you need things like 4E-BP1 and P70S6 kinase, or P70S6K to be activated. These eukaryotic initiation factors, these eukaryotic initiation factors work to create the ribosomal, the active ribosome unit. All right. So they do a pretty good job here showing that the ribosome is 2 subunits. There is a larger subunit and a smaller subunit. They actually determine that by looking at the molecular weight of the actual protein. So there's what's called an 80S subunit and a 40S subunit. And when they get together, they don't add up to 120. They actually make a 60S subunit because that's how much it weighs when it gets together. Okay. So you have these ribosomes. The ribosomes can come together because different eukaryotic initiation factors that are associated with the 80S subunit and the 40S subunit work to bring those two things together to make the functional ribosomal unit to get translation. Okay. So how can mTOR be activated? Let me count the ways. So we have IGF-1 and insulin. So growth factors like growth hormone, which leads to the release of IGF-1, for example, or an increase in blood glucose, which causes a release of insulin from the pancreas to facilitate glucose uptake. These are important signals that the system is not exercising, that nutrient status and energy status is high, and energy demand is low. So when energy demand is low and energy status is high, it's time to make hay, right? So these are anabolic processes. These are the conditions ripe for anabolic processes. So we're going to take advantage of those situations by stimulating protein synthesis, stimulating glycogen synthesis, stimulating fat storage, suppressing fat breakdown because we have carbohydrates in the system and that's our preferred fuel source, right? So that is a preferred fuel source over fat when the two are both in the system and available. All right. So we have insulin and IGF-1. Insulin and IGF-1 bind to the insulin receptor or insulin-like growth factor receptor. So these are receptor tyrosine kinases, RTKs. We talked about these very early in the term. They are trans-autophosphorylated, and then what they do is recruit these proteins that form this scaffolding complex, and they are activated through physical interaction with the receptor. So the receptor becomes activated, then IRS1 becomes activated, then it activates PI3 kinase. Have I showed you guys the insulin signaling animation in this class? All right. Stick with me. How can I find it? Macs are the best. Do you know how long that would have taken on a Windows machine? Is the search gotten better in Windows? Whatever we're up to now? Windows 20? Nobody knows? Come on, you guys are in the game. You use computers every day. Okay, so this is a slide that I made when I was in graduate school for my master's thesis. So my master's thesis was based on trying to understand sensitivity to insulin signaling. So one of the things that we know when people lose their insulin sensitivity, like in the case of type 2 diabetes, they actually still produce a lot of insulin. They still express a lot of insulin receptors. Insulin and the insulin receptor still bind together efficiently, but there's a breakdown in communication in these intracellular signaling pathways. And so what you can do is take a look through taking muscle biopsies in insulin-activated tissue and find out like where is the breakdown occurring, right? So this is one of the examples. Now this example is referring to how we get the intracellular protein GLUT4, which is a glucose transporter. How do we get this from inside of the muscle cell to the membrane of the muscle cell to allow for glucose to get into the muscle cell in the postprandial state, in the after eating state, right? So again, the conditions here are anabolic conditions. We have, we're at rest. This individual is at rest. They just consumed a meal that's high in carbohydrates. So they have a rise in blood sugar, low energy demand. That is going to initiate the pancreas to release the hormone insulin. And then insulin is an anabolic hormone that's associated with anabolic processes, things like protein synthesis, blood glucose uptake, glycogen storage, fat synthesis, suppression of fat breakdown. Okay? So those are all things that are associated with rest and recovery. So here's the insulin receptor. This is the receptor tyrosine kinase that was in the figure. This is similar to what's in the figure. So we have insulin that comes down and binds to it, and that receptor tyrosine kinase gets phosphorylated. That's what these little dots are. And what happens is that when insulin binds to the receptor and the receptor comes together, that's a process called dimerization. And when dimerization occurs, then these two sides talk to each other and phosphorylate each other. Once you have an active insulin receptor, then you can recruit IRS1. And it gets phosphorylated on specific residues, right? So, and we already talked about how depending on what residue gets phosphorylated, proteins can be activated or deactivated. So in this case, this is getting phosphorylated on threonine residues, which actually activate it. Then it recruits this protein. This is PI3 kinase. PI3 kinase has two subunits, and so I've labeled those here instead of just saying PI3 kinase. Because there's a regulatory subunit called the P85 subunit that binds to IRS1, and then it activates what's called the catalytic subunit, or the P110 subunit. That changes this membrane-bound protein, which you don't need to know the full name of. It's phosphoinositide phosphate 2 to phosphoinositide phosphate 3. So it adds that third phosphate. That's why PIP2 goes to PIP3. Now, activated PIP3 can move along the membrane and interacts with PDK1. And PDK1 then can activate AKT/PKB. Now, in the book, this AKT is called PKB, but they're the same thing. In the cell signaling slide that we looked at, it's called AKT. Now, in this scenario, AKT goes on to activate AS160, which is a subunit that's activated by GLUT4, and then glucose can enter the cell. Okay? And this is how insulin signaling works, and this is how cellular signaling works for the most part. Now, what's important, there's two things. AKT does a bunch of other stuff too, right? what is AKT also activating during this time? What intracellular protein that's really important for turning on protein synthesis? mTOR, the one we've been talking about, right? So in the summary slide that we were just looking at, we're looking at this same pathway, but not in the context of lowering blood glucose. We're looking at it in the context of turning on protein synthesis. But if there's low energy demand on the cell, and there's a lot of glucose in the cell, that's the perfect time to turn on protein synthesis. So it makes sense that insulin would do both. All right, so glucose comes in the cell. We have a high energy source. We have a high energy status, low demand, and now AKT can go do a bunch of stuff, right? And AKT is going to activate mTOR, and then mTOR is going to go do a bunch of stuff. It's going to turn on protein synthesis. It's going to turn on PGC1 alpha. It's going to turn on all these different things, right? Now, this is just one way to turn on anabolic signals. And that's what that figure that we were just looking at is showing, where this is one way to turn on an intracellular signal, but there's other things as well, like the resistance exercise itself, the physical movement of it can be sensed by the cell, which can also turn on mTOR. The presence of amino acids can turn on mTOR. Now, what's interesting is that we talked about what was the main transcription factor that turns on the mitochondrial biogenesis, PGC1 alpha. It was one of the main ones that we talked about in the last section when we talked about cardiovascular adaptations or endurance adaptations. It's a result of the Lohmann reaction. What gets activated? AMP kinase. So AMP, great job. So AMP, right, 2 ADPs, there's a rise in ADP because of regular skeletal muscle contraction. 2 ADPs get together and make an ATP and an AMP. The AMP activates AMP kinase, and AMP kinase turns on a whole bunch of transcription factors that are related to endurance adaptations. Now, what's interesting is that in the absence of insulin, so when we're exercising, is insulin going to be present or not present? Not present, right? So insulin goes down. But blood glucose still goes up. Your liver starts dumping out blood glucose. You may be taking on some Gatorade and your blood glucose goes up, but you're still running. So how do we get blood glucose into a muscle cell when there's no insulin to drive this process? Right? Because we don't want this. This does so much more than just get blood glucose into the cell. It also turns on all these pathways that are associated with rest and recovery. But if we're exercising and we still have a need for glucose, but we don't want to turn on anabolic pathways, how do we do that? All right? And so this little guy here, AS160, in the absence of insulin, AS160 can be turned on by AMPK. And this is called contraction-stimulated glucose uptake. So through muscle contraction, we get a rise in ADP. The 2 ADPs come together to form an ATP and an AMP. The AMP activates AMP kinase. The AMP kinase activates AS160, and AS160 can recruit GLUT4 to the cell surface just like it does in insulin signaling, but in this instance, it's insulin independent. And that's how we get GLUT4 to the cell surface during exercise. So if you were a type 2 diabetic and you were resistant to the action of insulin, what's the best way you can lower blood glucose levels? Exercise. Right? Because then you can activate, and in fact, the most commonly prescribed type 2 diabetic drug is a drug called metformin. And metformin is a pharmaceutical activator of AMPK. So you activate AMPK, It activates AS160. AS160 recruits GLUT4 to the cell surface, and you can lower blood glucose. Okay? Of course, when I presented this in my dissertation, my advisor goes, "That's a pretty cool slide, and I'm sure you spent a long time on it, but that person is now deceased because of low blood sugar. So could you redo it and leave a few molecules of glucose up in the bloodstream?" I said no. It's pretty... Slide is famous too. And when I say famous, I mean not famous. But I was at a conference last year, and because the professor that gave me crap about, he's a good friend of mine now, or he was a good friend of mine then too, and he still uses that slide to teach insulin signaling in his classes. And I was at a conference last year, and I met a recent graduate from my alma mater, and she was like, "Oh, I knew it." She was looking at my name tag. She goes, "I knew your name sounded familiar." She goes, "Did you make that insulin signaling slide that Dr. Melby always puts up in his classes?" I said, "Yeah." Good story, huh, guys? Thanks for the feedback. Okay. So now, in a less sophisticated way, we look at this still photo. But still, we have our insulin signaling pathway. We have our IRS1. We have our PF3 kinase. We have PIP2 got changed to PIP3. We have PDK1 that turns on PKB, which we call AKT. All right? And then in this instance, now we're interested in how AKT controls mTOR and how mTOR controls protein synthesis, because that's the major adaptation to endurance training, or to resistance training. All right? And so what's important about this, is that we get adaptation in response to resistance training, and insulin can be a big part in facilitating that, which is why a post-exercise drink shouldn't just have amino acids, it should also have some carbohydrates. Does that make sense? Because you want to turn on insulin, and you want to turn on protein synthesis via the insulin signaling pathway, and you want to turn on protein synthesis via amino acids, and you want to turn on protein synthesis from the actual exercise itself. The other thing this is showing, so if you see here, AKT blocks TSC2, which then relieves the inhibition on mTOR and allows it to activate. AMP kinase does the opposite. It phosphorylates TSC2, which means it's going to do what to mTOR? Turn it on or turn it off? Turn it off. Right? And this is why mTOR is thought to be unique to the adaptation of resistance training and not as associated with the adaptation towards endurance training. Now, I'm not totally convinced that you don't get any mTOR activation when you're not endurance training. Does that make sense? So I still think when you're done doing endurance training, you get some activation of mTOR, which leads to increase in protein synthesis. So your carbohydrate and amino acid drink is important. But why is mTOR, which is regulated to protein synthesis, it's turned off during endurance exercise because AMPK is up during endurance exercise and can prevent mTOR. All right? Does that make sense? What? What are you thinking?
A lot.
Okay. But this is why, when you look at the literature, there's a field of study that I've mentioned several times called concurrent training. Concurrent training is when you're training for sports like basketball that have an endurance component and a strength and power component. And you have to figure out how to optimize your ability within both of those categories. But those categories, from an adaptation standpoint, can work against each other. So you can reduce the effectiveness of a weight training session if you go and run right afterwards. Do you understand? So like the current working recommendation, the hypothesis in terms of optimizing training is that you should train your endurance component first and then wait 5 or 6 hours until AMPK has done its thing. And then you do your weight training next and take your recovery drink and get mTOR going. So that then you get adaptation to the aerobic side of things and you get adaptation to the resistance training side of things. Right? But you don't want to do those, you don't want to do the work, sorry, I don't want to say such definitive statements, but the current suggestion is that if you say lift and then you do your cardio afterwards, that you're negating some of the benefit that you just did in terms of strength and adaptations to resistance training. Because you're turning on AMPK and you're preventing the activation of mTOR. Questions on this? Thoughts on this?
So like the endurance part, since it turns on AMPK, but if they were to take a recovery like amino acid drink after, does that then turn on the mTOR so that they can adapt in that way?
Yeah, so this is a really great question. So from a protein synthesis standpoint, there are also other things that can activate protein synthesis and activate P70S6 kinase and those types of things. This is just one of those ways, right? Now, AMPK is going to be in the system, but I actually don't know what the half-life of AMPK is. So like, if I go for a 5-mile run and then I stop, I don't know how long AMPK is like in the cell, activated in the cell, like activating some of those transcription factors for things like mitochondrial biogenesis and those types of things, and turning on endurance type adaptations.
Yeah.
Does that make sense?
Yeah.
But you still get a rise in insulin, and insulin is still a really powerful anabolic hormone. There's more things that are activated off this insulin receptor than just IRS1 and PI3 kinase. There's MAP kinase pathways, there's other mitogenic pathways that are associated with anabolic processes that insulin turns on. So I still think it's really important to get the post-exercise recovery drink after endurance training in order to get those adaptations. It just might not be as high a level of mTOR1 association and activation as in the post-resistance training state.
Okay.
But the protein synthesis is still stimulated in an insulin affected way in response to endurance training.
Okay.
The other thing about taking on a high carbohydrate drink in endurance training is not only does it turn on insulin, but what else does it do? It starts to replenish what? Glycogen stores. Right. So all these things, right? All these things that we learn about in binary terms. We learn about glycogen. We learn about energy. We learn about protein synthesis. We learn about amino acids. We learn about all these things are, it's like your body is not wasteful. It's really smart in how it uses these signals. Right? So we're trying to cross that threshold of, all right, I know a bunch of stuff individually, and now I'm trying to push you to see how all these things come together. Right? And we're kind of getting out there now. Like, you're starting to ask questions like, how long does AMPK last? I don't know. I mean, somebody knows. There's a woman out of Harvard named Lori Goodyear. She spent her whole career studying AMP kinase. So only protein that she studied, she's probably published 50 papers on it. So I'm sure she would know. There's a guy out of UC Davis named Keith Barr who studies all this type of molecular signaling. He's a really bright guy and writes a lot of really good articles. He has some of the most, what I think are the best written articles on concurrent training. It's where like some of my interpretation and opinions have been influenced by his writing. And he has suggested that it's 3 hours. The window is 3 hours. So if you weight train and then you want to get some cardio in, you need to wait at least 3 hours. If you do it before that, then you're taking away from your weight training adaptation. And then there's also some really cool articles written about some really early exercise science articles. One of the fathers of exercise physiology is this guy named Costel. I forget his first name. Anyways, my master's advisor was trained by him. He was one of the first people to have a dedicated exercise physiology department and lab. It's at this small school in Indiana called Ball State University, which turns out to be like one of the founding places of modern exercise physiology. So the Ball State PhD class reunions are the stuff of legend. So you guys think I'm joking, but I'm not. That's what makes it so fun. Okay, so so there's a guy there that when he got there, he was an Olympic lifter, but everyone there was a lot of exercise science people are like endurance athletes, and so he started like running and stuff like that, and then he started losing all his strength, or he started compromising his strength, and he really got interested in this idea of like, why does these two things that we really need, why do they seem to take away from each other when we try and have both of them to the maximum amount? Does that make sense? And from your body standpoint, it would make sense to kind of be a jack of all trades from an evolutionary standpoint. It wasn't worried about winning gold medals in the power in the snatch, right? It was worried about being able to run when it needed to run, get away from things when it needed to get away from things, and get food when it needed to get food, right? Okay, so here's another, I think, really nice summary of mTOR. This is from a journal that's available to all of you through the library called Nature Reviews. Remember, a review article is an academic article that takes anywhere from, usually it's a minimum of 50 citations, can be up to 250 citations, and someone will read all of those, analyze all of those, organize them, and then write about the current state of the field, right? So that insulin signaling slide that I showed you, it wasn't like one experiment discovered all those different steps. It was one paper that would introduce one of those aspects, and then another paper would confirm it, and then another paper would introduce the next aspect, et cetera, et cetera. And over a 25-year period, we can then construct a PowerPoint slide that would give us some idea of what the hell is going on. See what I'm saying? So reviews are this really useful tool for practitioners and students. I read reviews all the time, still to this, whatever. I read reviews all the time, and it's because if I want to know something about mTOR, you just go and get the latest review on it. And then you have the latest 150 articles all compressed into one, 15 or 20 page paper, and you can get a lot of information in a short amount of time doing that way. It's very efficient. You also have enough information from individual studies that people model. Individuals can make a model and make these types of summary figures, right? So this summary figure is based on years and years of research. But we have all these common things that we've talked about. We have insulin, IRS1, PI3 kinase, PDK, and AKT. We have PIP2 going to PIP3. We have amino acids, and there's even more detail here, right? So in this slide, we have amino acids. Sorry, there's not more detail. Amino acids activating this protein called RAG, and RAG activating mTOR. We have glucose directly affecting things that affect TSC1 and 2, which affect mTOR activation. We have our genes in here and all the different transcription factors that are activated under different conditions and going in. And again, you're not expected at this level to memorize each one of these different proteins and figure this stuff out. The point is, though, is if you're a coach, if you're a personal trainer, if you're trying to help somebody in a specific situation, you have some molecular tools in your toolkit. So you can understand some complex physiology, and you can go to an article like this and go like, all right, this starts to sound familiar. I'm just going to start at the top, pick this apart piece by piece, and see what I can put together and understand. Okay? And that can be insightful when you're doing work. Okay, so that's mTOR. Okay, next thing we're going to talk about is myostatin. We've already talked about this a little bit. Myostatin is a protein that is a brake for muscle growth. So it has the opposite effect that the mTOR pathway does. Whereas mTOR pathway turns on hypertrophy, myostatin suppresses it, right? So when we have animals in which myostatin is knocked out, they have significantly more muscle size than animals with it. Myostatin functions through an intracellular pathway called the SMAD pathway. By the way, if you discover one of these proteins and basically say this protein exists in the cell and it does this, you get to name it whatever you want. So there's all these, a lot of times these pathways have acronyms that I a lot of times don't even know. When I say stuff like that, I'm not saying that I know everything. I'm just saying, like, even somebody that has looked at, I'm just thinking about how that sounds when I say, even I don't know. Like, I don't know a lot of shit, okay? So I don't know what SMAD stands for, but it stands for something. There's also a Sonic the Hedgehog protein, I swear to God. Because the dude that invented it, his kid was super into Sonic the Hedgehog, the Sega game. You guys know what I'm talking about? Yeah, okay. Okay, so when myostatin is translated into a protein, it's not functional. It has this really unique activation sequence. All right. So again, we've talked about how just because a polypeptide is made and a protein is there, it doesn't necessarily mean that that protein is functional at that moment, and myostatin is a really cool example of that. So when the myostatin protein is turned on, the full-length myostatin gets cleaved, so the inhibitory part of it gets taken away, and then the activated part of it gets activated. So it's activated myostatin's effect occurs on a receptor on a cell surface receptor that is outside of the cell. Right? So the myostatin protein is inside the cell and it's in its full length non-functional state. It gets activated, it gets cut into its functional state, then the functional myostatin has to leave the cell and turn right back around and bind to its receptor. So activation of this receptor leads to the activation of the SMAD pathway. And when you have phosphorylated SMAD, it leads to the activation of SMAD4. That SMAD4 is a transcription inhibiting factor. Right? So it's a nuclear protein that can go into the nucleus and suppress transcription. And if we suppress transcription, then we have less mRNA. If we have less mRNA, then we have less translation. If we have less translation, then we have less protein. If we have less protein, we have less of a phenotype. Okay? So, let's think about this. You do some work, you get some activation, but then all of a sudden, myostatin says, hang on a second, you're getting way too swole. So myostatin is formed. You have a full-length protein that gets activated by proteases. Proteases are just enzymes that break down proteins at specific amino acid junctions. You get the functional myostatin, which is this really small protein, 24 kilodaltons. I'm sure that means a lot to everybody. It leaves the cell and sheds its inhibitor proteins so that then it can be activated and bound to, it can be further regulated outside of the cell. All right, so this is one of the ways that you can fine-tune exactly what you need in terms of myostatin. So you can make a bunch of it, you can cleave a bunch of it, you can send a bunch of it out into the bloodstream, you can modulate how much of it gets bound to these activin receptors. The activin receptors activate SMAD2 and 3. SMAD2 and 3 activate SMAD4. This forms a complex that goes into the nucleus and prevents growth. All right. And the other thing you can guess, I don't know this, but I would bet if I had to that myostatin does a lot more than just what is shown in this slide, which is why it gets released from the cell. If myostatin is getting released from a muscle cell and myostatin is activated under particular conditions, then that myostatin out in the bloodstream is probably going to other tissues and telling them what the hell is going on at the skeletal muscle. This is how tissues communicate. Right? So all these signals, and can you imagine all this stuff is going on like all the time without even, we're just sitting there thinking we're being unproductive and doing nothing. But really we got all this SMAD activation and myostatin in our system and just rocking and rolling, man. Never complains, just chugs along, always shows up. How cool is that? Okay, so if we look at the myostatin gene, we can get wild type whippet. This is a certain type of breed of dog. It's often it's kind of like a mini greyhound, I guess. They race whippets a lot. I don't know if dog racing is still legal, is it? Okay. Anyways, so this is a wild type whippet. This is a heterozygous whippet, so you get about 50%. And then this is the full myostatin knockout. All right. This is the myostatin knockout bull, which we've looked at before. Right. So now, where can you see the application? Where can you see the application of something like this? Something like myostatin knockout. Who would this be of interest to?
People who produce food.
What?
Like, oh, never mind. I was just saying, like, people who grow.
Oh, they have no body fat though.
Yeah.
They have really high muscle metabolism, and they have really low body fat. So it's an issue in terms of marbling the meat. I don't think it makes good meat. I could be wrong. What?
You say like chicken though.
I don't know. It's a good point. What else from a non-food industry perspective? Yeah, so this is great. So it's not just people that want to get swole, which they want to find ways to modulate myostatin to get gains, right? Like they're looking for like drugs that can either suppress myostatin or even like alter myostatin regulation so that they can slow down this break and get more muscle mass, right? But if you think about it in terms of this condition called muscle atrophy, so this is uncontrolled loss of muscle in response to certain diseases or certain injuries. So for example, people that undergo, that get large burns, large and severe burns, one of the biggest issues that prevents them from recovering is that they have this massive loss of muscle mass that cannot be explained from a nutrition standpoint. So even when they're fed or tube fed or intravenously fed with the absolute ideal ratios of amino acids and calories and all this stuff, they still just like waste away. they go into this really inflammatory state, right? There's other conditions like muscle dystrophy and Lou Gehrig's disease, and cancer. Cachexia is a condition of muscle wasting that a lot of cancer patients see. So if you could modulate this in a way that could help preserve some muscle mass, even just general aging, to maintain functionality in people that have become wheelchair bound and those types of things because of loss of muscle mass. So you can see there's some pretty important, potentially important therapeutic implications to understanding this. And when we talk about the neuromuscular activation of skeletal muscles in a couple weeks, one of the things we're going to talk about that's interesting about these mice is that even though they will sometimes have skeletal muscle masses 2 to 4 times higher, they do not have 2 to 4 times the increased strength. Right? So just because you have a lot of muscle doesn't mean you can recruit all of that muscle in a coordinated fashion to produce power. And so a mouse with 4 times the amount of muscle mass, a myostatin mouse with 4 times the amount of muscle mass may only have twice the amount of predicted power. Okay? So we'll talk about why that is and some of the aspects that go into that. Thank you.